Welcome to the 9th talk of the webinar series on genetics. We are pleased to deliver today's talk. Professor Desai did her PhD from the Department of Molecular Reproduction, Development and Genetics at the Indian Institute of Science, Bangalore. After her PhD in 1999, she joined the National Cancer Institute, NIH, Bethesda, as a postdoctoral researcher. She was subsequently a research associate at the Division of Johns Hopkins, Singapore, from 2005, and a research scientist at the Genome Institute of Singapore from 2006 to 2011. She moved back to India and is an associate professor now at the National Institute of Biomedical Genomics, Kalyani. Professor Desai's lab works on data and applied using genomic and epigenomic tools to discover targets and to design diagnostic tests for breast cancer. She is a recipient of the Technology Transfer Award from the Center for Cancer Research, NIH, long-term fellowship to the Czech Republic from the INSA bilateral program, and short-term fellowships to the University of Department of Health Research in India. She is also a member of the editorial board of the Journal of Genetics. Today, she will talk to us about the evolving non-coding genome and its relevance to cancer. Kartiki.
Thank you, organizers, for giving me this opportunity and a platform to present our work. Thank you, Dr. Vidya, Dr. Prasad, and Professor Ranganath for inviting me. What I'm going to do today is talk about a different topic than what has been discussed in this series, because as Professor Vidya suggested, I'm a cancer biologist by training. What I'm going to talk about is the evolving non-coding genome and its relevance to cancer. So we are going to do a journey inside the cell, and just like in ecological niches and in evolution, you have push and pull and interaction between different constituents of that environment. We have the same thing also happening within our own cells. So I will begin a little bit in two parts. I'm going to spend a little bit more time on introducing the topic of non-coding genome, so that we have all our friends who are not exactly in the field of cancer come with us. And then in the second half, I'm going to summarize some of the data that we have been generating about a linked RNA. So what we are going to see is that a long time ago in the 1960s, the DNA which did not encode for proteins was considered to be just junk DNA. Okay, so it was not a bad way of calling it, because at that point what it conveyed was that there was little to no selective or determining advantage of this DNA to the organism, even when the organism was under selection pressure. And what eventually turned out to be a component of the C-value paradox was that the genome size of an organism and the amount of the non-coding DNA, they did not really correlate with each other, nor did they correlate with the complexity of the organism. What we mean by this is a small protist like amoeba can have 1 billion bases in their genome, and so do we. And humans are highly complex multicellular organisms, and we have 3 billion bases. So if you consider the equivalence between the genome size and the amount of non-coding DNA we have, there is really no correlation. Eventually, this junk DNA, which was coined by Susumu Ono, was then relabeled and recoined as junk DNA to describe the human DNA that was outside of the genes, because we know genes code for proteins, and whatever is surrounding was considered to be the junk DNA. But upon the publication of the human genome, we were a bit surprised that it revealed that there were far fewer genes present which produce proteins in our body than that was what was estimated. So we have about 20,000 proteins that are encoded by our genome, so largely remaining part of the human genome we can consider as junk DNA. But however, there was a group of scientists, and there's a lot of effort by other groups also, to give a function to this junk DNA or the non-coding genome. So one of the examples I've cited over here is the ENCODE consortium, and ENCODE consortium eventually by 2012 estimated that 80% of the genome actually may be functional, and it was not really useless. And its mission is still ongoing to find out the function of every one of the 3 billion base pairs that we have in the genome. So what it showed us by the DNA sequence or the genome sequence, human genome sequence, is that most of the genome that we have is non-coding. And what we see is the junk DNA is almost 97 to 98%. And protein synthetic DNA was only 2 to 3%, which encoded our disease. So what exactly do these non-coding DNA sequences do? So these non-coding DNA sequences, now as we are doing more and more work on these, we realize are constituted by multiple different types of entities. They could be transposons, they could be repetitive sequences or pseudogenes, and the ENCODE consortium is involved in finding all the regulatory regions or regulatory sequences that actually lead to regulation of the 20,000 proteins which are giving us the phenotype. A large part of these non-coding DNA sequences are also encoded into RNAs or ribonucleic acids, okay? So they are collectively termed as non-coding RNAs. They could be transfer RNAs or rRNAs, which are so basic to the functioning of our cells. And what we know now from transcriptomic data is about 75% of the genome is actively transcribed to generate these non-coding RNAs. What we will see is that apart from this tRNA and RNA, there is a whole amount of non-coding transcripts, and I will describe them in the next slide. So certainly, they are present and they are going to achieve some amount of functional complexity. So what we think now is that although we have only 20,000 proteins, the functional complexity that we achieve as a human being largely comes from the regulatory regions, or the way there is an interplay between these non-coding sequences and the expression of these proteins. And on more on an evolutionary scale, we can consider them to be, they're thought to be the prelude to variation, because they can accommodate a lot of mutations or sequence changes without, for example, a pseudogene can get many mutations without having a functional consequences in the organism. Exactly the reason why the DNA was considered to be junk DNA before. There are also some reports, and there is a thought that these kind of sequences or the non-coding genome actually is a prelude to formation of newer genes, and eventually over many thousands of years might lead to evolution. So as I mentioned, there are multiple kinds of non-coding RNA. There are far more than the protein coding RNA genes. What we will talk about is not the tRNA or the rRNA, but another group of non-coding RNAs which are collectively termed as the long non-coding RNAs. The only reason they are called long non-coding RNAs because most of you know that the short non-coding RNAs are about less than 200 base pairs long, and anything that is not making a protein is above 200 nucleotides is termed as a long non-coding RNA. And these long non-coding RNAs on an average can have large transcripts as large as 10 kb. And they come from various regions of the genome. We will not get into that right now. So just like we thought that the DNA, which we could not find a function to very early, was junk DNA, these RNAs were also considered just to be transcriptional noise. Now, where did the noise come from? It was largely from the transcriptome or the RNA sequencing data that was carried out from multiple tissues, organs, or disease phenotypes or cancers, where you found that a large part of the RNA that you sequenced did align with the genome, but it did not necessarily align with the protein coding regions. So where did this noise come from? So in early days, people said this was just noise, and these transcripts were expressed because they were just examples of low fidelity binding of RNA polymerase to scattered sequences or promoter-like or AT-rich sequences which are present throughout the genome. So eventually they just were made by chance. Okay. However, what we observe over the years is that the number exceeds that of mRNA. The link RNAs are made in huge amounts, but each of the link RNA was expressed at very, very low levels. So it took really deep genomic and transcriptomic sequences to identify these link RNAs, where by then you had discovered all the mRNAs that were present. So because you sequence so deep, there is a chance that your noise will amplify, and these went into the box of transcriptional noise. The other important consideration was that, as you see, the protein genes are very well conserved in function. You can start with Drosophila and all the way to the Eutherian species. You will find that the protein, which is a kinase, has a particular domain and it always functions as a kinase. For example, a protein that goes into the membrane always has similar kind of structure or some sequence. So, unfortunately, the link RNA across species show very low conservation. Okay. The third thing is that unlike protein sequences which are highly controlled and do not show very high rates of sequence change, link RNAs are known to change very frequently or at a greater speed. If you calculate this rate, that is the number of changes in a given link RNA sequence, and divide it by the number of changes in a sequence of the same size, that is under no selection pressure, for example, a transposon sequence, then you will find, and then if you convert the rate of transposable elements to 100%, you will see that the change in protein usually ends up only 10%, whereas the link RNAs show variation as much as 95% in their sequence, depending on the type of link RNA you are studying. The other thing, unlike a genome size, which was the C-value paradox, you could see that as the complexity of the organism increased, the number of link RNAs that you find in the genome appeared to increase. So on some level, people did not give up and started characterizing exactly what this transcriptional noise was. Like I mentioned, link RNAs have very low sequence conservation as compared to proteins, but somehow these link RNAs display very high level of tissue specificity in gene expression. So I've just taken an example from the GTEx data over here. As you can see, a particular link RNA that I'll talk about later, Hotair, is very highly expressed in the arteries, but you can see most of the organs and the brain, there is hardly any expression of this link RNA. It's barely detectable. So in this manner, multiple of these link RNA shows tissue specificity, and they are not really conserved across even human and mice or voles, and they show differing lengths, different loops, though overall structural some amount of similarity you can see over here. So as of 2018, the noise constituted more than 200,000 link RNA transcripts that were found in humans. If you look at sequence conservation across like human, mouse, rat, so on and so forth, you will find that there was high level of sequence conservation only in 3,000 of those link RNAs. So most of the others were not conserved. Now what we don't know and what is still a point of debate is whether the sequence of RNA is not important, whereas the structure of the RNA is important, and therefore you might be having lack of sequence conservation, but maybe the RNA folds into a similar type of structure inside the cell. That means its secondary structure is the one driving its function rather than its primary structure or sequence. Now, we don't know really if that is true or not, but we know there are lots of link RNA transcripts, and on some level, we have to find out if it's just noise or if they really contribute to any function in the human or even in disease phenotypes. So the reason people remained interested in link RNA, because as you took one link RNA at a time, we started discovering functions for them. So broadly, you can classify what the link RNA does into three types. The first one, it acts as a guide. Very often people see that this link RNA can bind something like a transcription factor and direct the positioning of the transcription factor on the genome such that it can regulate its target genes. The second is just scaffolding. Because the RNA can twist into different structures, it is thought that the RNA binding proteins actually bind different regions of the RNA, and because the RNA folds, they come in context of each other and show protein-protein interactions. So basically, this can act as a bridge between two proteins and probably lead to some kind of function. The second thing might be that which is also seen is exactly opposite. Instead of promoting protein-protein interactions, it can also act as a squelching agent. In the sense, the link RNA can take away one partner by binding to it and prevents the interaction of this green protein with its gray counterpart. And this may inhibit also the protein-protein interactions. Again, you go via scaffolding. The third is decoy, which is very interesting idea, and people have already shown this occurs. Now here we come to the junction where the messenger RNA, the short non-coding RNA like miRNA, and the long non-coding RNAs come into context. We know that the miRNAs bind mRNA sequences and prevent translation very often in the cell. Now, if you can imagine a link RNA which is homologous to the miRNA, what it does, it acts as a sponge. And by doing a sponge, it competitively binds this miRNA. So now it is going to partition the miRNA that is expressed in the cell between itself and its target messenger RNA. So very often in this way, not only it acts as a depot for the miRNA sequences, it can also prevent or positively regulate translation because it removes the negative regulator, which is the miRNA. So it forms a decoy sequence. It looks exactly like the target, but it is not the target of the. So as we did cancer sequencing, as you know, there's a lot of effort in sequencing cancers by huge consortia, which are all over the globe. And when they sequenced the whole genome and analyzed all the transcriptome, that is the RNA data, they found that several of the I'm going to switch to cancer over here, is that a lot of rearrangements in the genome also occurred in the link RNA regions. Then many single nucleotide polymorphisms that associated with cancer susceptibility fell into the regions of non-coding genome, and particularly that of the link RNA regions. Further, there were many mutations that were found to fall into the link RNA region, and as people did candidate approaches and tried to characterize these mutations, they found that they affected the link RNA function. What people then did was they also found that when you compare normal versus tumor tissues or different tumors from origin of different organs, there was a differential expression of these link RNAs in cancer tissues. Now, if you clone this link RNA and you force its expression ectopically or exogenously into a healthy cell, some of these link RNAs were capable of transforming that healthy cell into a tumor cell. So you can see that just like a protein was known, or a mutation, for example, in an oncogene could, or overexpression of an oncogene could transform the cell, just expression of this RNA could start the program of carcinogenesis. Now, having done that, this is a highly complicated figure. The point that I want to make is that characterization of individual link RNAs show that link RNAs are functioning almost in every hallmark of cancer, whether they are promoting genome instability, they're promoting metastases, they're getting involved in proliferation, and they're also getting involved in growth suppressor functions, so on and so forth. So they have a large impact on the function of and the establishment and progression of cancer. So how did we get into this field? Actually, we got into this field very serendipitously. We did not have an intention to go into link RNA biology when we started. And as Professor Vidya suggested, we largely are a group which is interested in breast cancer biology. And we work at the zone of breast cancer where we want to find options for women after first line of therapy, because you know now a large great and efficacious therapy is available for breast cancer. Most of the women do very, very well for 5 to 10 years on this therapy, and most of them remain in remission. Only a portion of them, like 30 to 40%, come back to the clinic. That means their cancer recurs, or they develop resistance to whatever they are treated with. So we have a goal whether we can find a biomarker or a molecule or a therapeutic target in these kind of patients, or if not that, can we at least assess which woman is not going to do well under the existing therapy by doing kind of genetic signature establishment or a biomarker establishment. So whilst we were doing this, we did a genotype to phenotype screen a very long time ago, and we discovered a protein called Jumanji domain containing protein 6, or for short, JNJ6. Now, this protein was very interesting for us as a target because apart from the biology, it is an enzyme. It modulates histone, it arginine demethylates it, it also can lysyl hydroxylate the histone, and it also has similar kind of enzymatic activities on major players in cancer such as estrogen receptor, P53, which is the famous tumor suppressor, and also splicing factors and histones. Now, interestingly, though it seems that this protein might be epigenetic modulator, it really does not have a DNA binding property. Counter to that, it actually has a single-strand RNA binding protein activity. And very recently, it was also shown to be a kinase. So usually, molecules or proteins that have so many enzymatic activities tend to be good targets for developing small molecule therapies. Having said that, I've just summarized a small portion. What we quickly found out after studying a lot of breast tumors is that GMJD6 was always associated with high grade of cancer. It was also associated with more ER negativity and in aggressive disease, which was stage 4 or stage 5. We went on to do a lot of cancer biology to understand exactly how GMJD6 works. I'm not going to go into that in detail, but what I would like to switch is to something else. So Ifang was a graduate student in my group. So this was a long time ago when I was in Genome Institute of Singapore. And I apologize for the next panel. I didn't realize it was so fuzzy. After doing all the characterization of GMJD6 before she submitted her thesis, she actually went ahead to find, because it is an RNA binding property, what kind of RNA interacts with GMJD6. So she performed an experiment called RNA immunoprecipitation followed by sequencing. So we have a specific antibody that is against our protein of interest, which is GMJD6. So if you take cells, GMJD6 is technically bound to the RNA that it interacts with. You cross-link the cells so that you preserve this RNA-protein interaction. Then you lyse the cells, get the chromatin out. Sorry, the RNA protein interaction out, and then you add an antibody to GMJD6, and you pull down. As you pull down the protein, all the RNA that is associated with the protein is going to get enriched. The remaining RNA, which is not with GMJD6, you can wash off. And then when you release the RNA from this complex, you get a group of RNAs that potentially interact with GMJD6. So how do you find the identity of this RNA? Is by RNA sequencing or by next generation sequencing. And then after you map the reads, you can find out. So we got a series of RNAs that were binding GMJD6. And there are many, we have not had time to go through all of them, but the one that caught our eye was Hotair. Okay, so it is a long non-coding RNA. The reason we got interested in this because Hotair mediates transcriptional silencing of the Hox C class locus. That means it had important activities during development. So Hotair is actually expressed from the Hox C cluster. The RNA then binds two protein complexes. One is the PRC2 complex and the other is the LSD complex. And together, this huge complex is directed by Hotair to go to certain regions of the genome, one of them being the Hox D cluster. What this complex does is puts a negative histone marks or negative regulatory marks like H3K27ME methylation marks such that the genes in the surrounding region are completely silenced. So this was the activity. So we estimated that if GMJD6 binds Hotair and GMJD6 is an epigenetic regulator and Hotair itself is leading to gene silencing, so GMJD6 in somehow must be coming in context with the PRC2 complex, so on and so forth. And if you can see human and mouse Hotair, this is the conserved molecule here. Unfortunately, as we were working with Hotair, there was a group who actually did a knockout for Hotair and showed that Hotair was really dispensable for mouse development. As you can see over here, there is not much of a difference between the Hox D gene expression in these embryos when you knock off delete Hotair, and you don't really see a staggering phenotype. So this was a bit unfortunate at that point, but then the jury is out because there is another paper which suggests that when you knock off Hotair, you actually get skeletal abnormalities. So that means because these are homeobox genes, HOX-C and HOX-T, they did give a homeotic transformation when in one study where Hotair was deleted and in another it was not recapped. Nevertheless, we went ahead with the JMJD6 and Hotair interaction, and what IFAN quickly found out was there was an increase in when you increase JMJD6, that is when we forced the expression of JMJD6 in cells that lacked the expression of JMJD6, Hotair immediately went up, okay, quite substantially in terms of, and this was done by real-time PCR or quantitative PCR. And on another level, we already did RIP-seq and got Hotair, but we validated it by doing an RNA-IP and doing PCR for Hotair and showed that the RNA-IP actually, they probably interacted physically with each other so that JMJD6 and RNA bind together. So then Antara joined my lab a few years later. She was my PhD student and she asked the question, what about JMJD6 and Hotair? Does JMJD6 affect transcriptional silencing by Hotair? Does it do it by interacting with the non-coding RNA and then affecting this complex formation and then interferes with the PRC2 mediated gene silencing? We know from others have shown that Hotair also interacts with this particular protein called EZH2. This EZH2 is actually a component of this PRC2 complex. Okay, it's one of the constituent proteins. And Hotair directly binds EZH2. So is the effect of JMJD6 and EZH2 together, and it is not really via Hotair? So she asked these questions in the lab, and I'll briefly summarize her findings in the next couple of slides. What she found out by a series of experiments, including Hotair promoter analysis, mutating that promoter, performing chromatin immunoprecipitations, using mutant JMJD6 proteins, and doing electromobility shift assays, that basically Hotair binds very close JMJD6R protein binds very close to the Hotair promoter and positively regulates it. Okay, so that's why we call it the proximal promoter. It's within 200 nucleotides that JMJD6 can bind. She went on to find the exact sequence where JMJD6 interacted with the Hotair promoter, and this is the sequence over here. Now remember, I will revisit this, that JMJD6 actually requires the DNA binding protein partner, because as I've mentioned, JMJD6 is not capable of directly interacting. So there is definitely a protein over here that binds, which also binds JMJD6. And that is a point of investigation by someone else in the lab currently. What Antara also went on to show by doing siRNA and overexpression experiments and following the RNA analysis, then doing molecular analysis, which details I will not go through, but I will summarize her work, and also using chip sequencing for both JMJD6 and EZH2, is that both JMJD6 and EZH2 actually co-regulated multiple genes as high as more than about 496 genes that were co-regulated by these two proteins. Most of the genes that were co-regulated were predominantly in the cell cycle pathway. They were targets of proteins which form the DREAM complex and the E2F targets. Now together, when you look, so from this we hypothesized that since both JMJD6 and EZH2 are epigenetic regulators, they must be binding upstream of these genes. So during our ChIP-seq experiments, what we found was actually JMJD6 definitely bound most of these genes in the upstream region or some genomic location close to it and regulated their expression. But we never found EZH2 being present in that region up to 100 kb around the transcription start site. So what we think in this paper, and we have not really proved it as yet, is that EZH2 is probably inducing another X factor, which is actually working with JMJD6 and probably co-regulating. And maybe this X factor and the protein interacting factor, which binds DNA, is the same, or it may be different. We have no idea. So what we summarized over here, though, in terms of breast cancer, is that JMJD6 and EZH2 are regulating the same genes but by different mechanisms. So when you do therapeutic intervention against only one of these targets, for example, JMJD6, you may not completely block the cell cycle because the activity of the second regulator will continue on the same genes. That means overall, the cells will progress into cell cycle. So what we proposed over here was that if you combine the drugs against both GMJD6 and EZH2, it might be of greater benefit and it might lead to better survival in breast cancer patients. So we talked about its functions so what she summarized, Antara, briefly in her thesis was GMJD6 appeared to perform independent functions, one with Hotair and one with EZH2. In terms of Hotair, it was only involved probably as far as we can see and in the data that we have, is GMJD6 induced the expression of the link RNA, and possibly this increased link RNA then feeds back and goes into this PRC2 complex and continues to work with this particular complex for gene silencing. In terms of EZH2, what we found was both EZH2 and GMJD6 promoted tumor cell proliferation, although by different mechanisms. And this function was independent of Hotair, because when we silenced Hotair in these cells, we could see that the cell cycle still progressed. So there was one way of feeding into the PRC2 complex, and there was an EZH2 function that was independent of PRC2 complex that GMJD6 and EZH2 commonly had, and together the net result was that it promoted tumor cell proliferation. So I have talked a lot about DNA regulation and regulation of Hotair itself, and also regulation of gene expression by GMJD6 and EZH2. So the question remains open. Like I mentioned, RNA and protein interactions were present between GMJD6 and Hotair. So what about those? Now, Hotair is a long non-coding RNA, so it's about 2.4 kb in length. There's a beautiful paper of chemical interaction. They showed that it is distributed into 4 domains that are given roughly over here as base pair sites, you can see. They each have their independent folding structures, and it is replete with multiple helical segments, loops, internal loops, and junctional regions, that all these are quite nice to do protein-protein interactions. So what we already knew about Hotair was is it 2.4 kb or 2.1 kb RNA, depending on which transcript you take. We knew that EZH2 binds the 1 to 300 region of Hotair already. We also know that LSD binds the tail end of Hotair. So we performed an experiment in which we used GMJD6 protein, which was purified, and asked the question, does it bind Hotair in vitro? So these are in-tube experiments. And we used the full-length Hotair in the beginning, and we increased the amount of Hotair in each lane. So you see the signal increases as we increase the RNA. And GMJD6 is a 55 kilodalton protein, so it appears along with the labeled RNA. Now, to show that there is some kind of finite binding, what we used was a cold competitor, which actually is the full length, which is not labeled by any by cyanine 5. So when you put it in, it binds and it removes of this signal. We also use actin RNA as a non-specific RNA over here because it should not be that GMJD6 binds all RNAs in the cell. So we wanted to show that it's reasonably specific to Hotair. So actin actually does not give us any signal, and even the H300, which is our construct, which is from 1 to 300, gives a band. So we know that it binds in the 5' end of the Hotair, and it also binds in the 470 region, which is the later in fragment 2, we have a region called H470, where we also got a positive result, suggesting that Hotair and GMJD6 also functionally interact with each other on the basis of RNA and protein. Now, this is just we have done. We've not gone further with these studies, but after looking at some of other preliminary data, which I don't show over here because it's really preliminary, we have a working model over here. What we are trying to ask over here, does GMJD6 and Hotair participate as an epigenetic regulator on its own promoter? That means the GMJD6 actually synthesizes the non-coding RNA, and this feeds back by binding to GMJD6 RNA and other regulatory regions. The reason we think about this is are multiple evidences, but also that the non-coding RNA function, this type of function is seen before with several non-coding RNAs in which, like I mentioned, they direct the protein binding of the transcription factor or the transcription modulator to binding sites. So we are thinking about this, and we are trying to design the experiments. I don't have any data to show you right now, and maybe next time we meet, I can give you some update on this particular hypothesis or this particular idea. So what we have tried to show is some amount of significance for what all this means, though it's a lot of molecular data, and it was definitely done in a lot of breast cancer cell lines, in some models, and in also using the publicly available TCGA data, and also by staining and immunohistochemistry of real breast cancer patients. So what we proposed in this study was that the high JMJ6 expression may be driving much higher levels of Hotair in breast tumors. What we know from our and other studies is that very high levels of Hotair actually promote metastasis and death. So we proposed over here that blocking JMJ6 in terms of Hotair may be useful in preventing the expression of Hotair and preventing such events which give worse prognosis to the breast cancer, to the woman suffering breast cancer, and poorer survival. So as you can see, when we use publicly available TCGA data, which has 817 RNA-seq profiles, we find that where only in tumors where JMJ6 and Hotair both are high, you can see the survival is worse than when JMJ6 and Hotair are much lower in expression. Okay? So we suggest that this kind of event may be taking place, and that blocking JMJ6 may be of value. So I've talked about only one non-coding RNA, which is linear. But now, as we sequence more and more, there is a huge effort in classifying all these non-coding RNAs. So we started simplistically with small non-coding and long coding, but you can see by last year, they have already been divided into so many different annotations. They could be small non-coding, then they are long non-coding. We talked about the linear ones, and we're trying to functionalize them. And as you can see, they go into various functions in the cell. But what we have not talked, I have not talked about other circular long non-coding RNAs. So these are extremely interesting, and they have their own biology and own way of doing things. We have not ventured into this in any way. We are sticking to the link RNAs, which are linear. And we are also actually embarking on a project in which we are going to try and functionalize a group of link RNAs that are associated with poor prognosis pan-cancer with my collaborators in NIBMG. So that also might be a topic that if we have some data a couple of years down the line to discuss as to what we find in terms of functionally annotated these link RNAs. So with that, I'm going to stop, and I hope I gave you some idea of a different field and interested, especially the young people, to go into this field. This work is not possible at all without a lot of collaborators, both in the hospitals like Tata Medical Center and Christian Medical College in Vellore. I also have a collaboration with Professor Kondaya, who is also my PhD supervisor. I retained the collaboration with Genomics to Singapore, though both Ed and Yifang have moved on to different institutes. And I have a very nice collaboration with the University of Chicago for doing the signature work, which I did not talk about today. And it's only possible with a lot of funding from various agencies, so I thank it. This is my current group. They're all my PhD students, and these are two past members of my lab. Unfortunately, I don't have a picture of Antara, whose data I already presented over here, so sorry, Antara, about that. So with that, I thank you for your attention. This is a rainy day in NIBMG at the main gate of NIBMG. I also invite you to visit us as soon as the lockdown period is gone and we are better off traveling, and we can go back to traveling. And thank you very much for your attention. I'll be very happy to take questions.
Thank you so much, Kartiki. There are a couple of questions I see,
Okay.
Probably not related to your cancer work, but Swati Adav asks, your comments on Google's DeepMind solving of protein folding.
Sorry, can you repeat that? You were breaking up.
Swati Yadav would like to have your comments on Google's DeepMind solving of protein folding.
Oh, okay. So Swati, thank you for your question. I'm only a card-carrying bioinformatician. I am probably not the best person to answer this question. I'm very sorry because I don't really do a lot of bioinformatics. I'm a very hardcore molecular biologist, cancer biologist. So, but what I can do is talk to my friends at NIBMG and try to get you back for the answer.
There's another question. What is the reason for the name Jumanji protein? It's a very interesting name.
Oh, I actually don't know the answer to that. I've hunted for it, but it came a long time ago because this Jumanji domain, it's a domain called JMJC. It is there in multiple proteins actually. A lot of them are these Fe2 based hydroxylases. So it's a family. So it is JMJC is a domain actually, and I think it came from Jumanji from that time somewhere, probably in Drosophila or something, and then it has stuck because now you have isoforms of those. So the origin is somewhere in the developmental biology, but I could not associate it with. I did look for it, the reason for that, but I couldn't associate it with the phenotype. It just begins with JMJC domain. So that's an interesting question, though. Even I'm wondering about that. Yeah.
And again, a question not related directly to the talk, but could you comment on the mRNA vaccine for COVID? There seems to be a lot of interest in.
Oh, really?
People.
What do you want me to? That's the comment I have, and well, I really don't have anything to say about the vaccine.
Can Hotair also transform when stressed?
So thank you for asking that question. Actually, there is another group actually who has tried also doing that. We also tried doing that, and it doesn't transform. It requires this PRC2 and this whole complex and everything, but it doesn't transform. Like that is why actually we were a bit disappointed when we knocked it off. We couldn't get any phenotype in the breast cancer cells, and then I found this paper which actually said that they didn't get a phenotype in mouse either. So yeah, on its own it doesn't transform. It requires another factor to be present. So it seems to be more like a link RNA which is fine-tuning something. A lot of link RNAs we find now is actually fine-tuning a more master regulator. So I think Hotair belongs to that class. So it didn't turn out to be an oncogene. But if you remove it, in some cell types, you will have loss of metastasis function, but not all. So that is why it came to be associated with metastasis actually. And if you just see the expression of Hotair and you do a correlation analysis with metastasis and death in breast cancer patients, you will see that mostly high Hotair correlates significantly with women who have died due to, who have lost their lives due to metastasis.
Pratibha Ranganathan asks, do you have experience of profiling circular RNAs too?
Okay, yeah. So circular RNAs actually easily enough, somehow in, when you get a lot of, you have to really do a deep transcriptomic data. I mean, see, I'm looking at it at a point of view from the genomic studies that we do. If you do deep enough, you get a lot of unmapped RNA actually. It is sequenced, but you cannot map it in any way to the genome. So a lot of these leftover RNAs is then mapped to, PV RNA, SNO RNA, the small ones, the big ones. And the remaining actually, if you circularize them, they then map, you can map them, then eventually you find out they're circular RNAs. So it is a very interesting way now. Because there are circular RNAs, there is a different way of making the library for the circular RNAs. But their expression is very, very low. So, Pratibha, I can send you if you're interested in the technique, I can send you how it is done, the library.
How can we see the future of cancer therapeutics in ncRNA, protein interactions, or ncRNA biology?
Okay.
And related to that, how far can we address the problem of resistance through this?
Okay. So I'll try to answer the first questions first. So we know universally that targeting an RNA is much more difficult, right? However, what people are thinking about doing is actually targeting the interacting partner. So that is why we got interested in all these interacting proteins and all. The second way people are trying to target is actually using a siRNA approach or miRNA kind of approach where you prevent the expression of this long non-coding RNA. Okay. The third way is that we know that a lot of these miRNAs are also bind link RNAs. The third thing that I showed you, the decoy. So they are trying to make decoys by using aptamers for miRNA and then using these aptamers to block the link RNA. So what link RNA was normally doing was blocking, was sponging off the miRNA from its function. You're going to use that as therapy in which you give an artificial RNA which is going to sponge off the link RNA. And people hope that these approaches will work in therapy. I'm sorry, the second question, the future of link RNA. Yeah.
How can you address the resistance through this?
So right now we are at the stage is that we have a lot of association studies, right? We know that if I compare breast tumors which are responsive or sensitive to a blocking agent and then we compare another tumor which is resistant, we get a lot of difference in these link RNA expressions. The question is, if I take certain of these link RNAs, not all of them, Hotair is a bad example. If I overexpress the link RNA, say in a heterologous system like a cell line, it does promote a resistance. Okay, so there is definitely some kind of some kind of relationship between resistance and link RNA. And going back to your first question, that might be the other way of blocking the link RNA again, if it is going to promote resistance in. See, the resistance is largely done, is shown to be by squelching. Okay, a lot of these link RNAs, because of their binding proteins, they are removing those proteins such that now the thing becomes resistant to the drug. So it's the same mechanism which we were thinking for protein-protein interactions, and disrupting protein-protein interactions could be one way of preventing resistance. The same way, disrupting a link RNA protein interaction might be a way of. But definitely we have some link RNAs which are overexpressed in resistant cells and they can confer resistance to cells which are not resistant.
Okay. I think there are no more questions. I'd like Professor Rangana, did you have some questions?
No. Kartiki, very basic question. In the beginning of the talk, you said the sequence could be the same, the RNA, but the structure could be different. But usually we say the structure and the sequence is correlated, but how could the same sequence can assume different structures?
Yeah, so this is the unique thing of RNA because it can fold back on itself because the RNA interaction will be on the basis of Chargaff's rule, which is ATGC content and the run of A's and T's. Right? What RNA can do is bulge off. So that's why if we, I'm just going to go back to this kind of sequence. So I'm just going to use Hotair as an example. As you can see, you find this stem loop kind of structure, right? If you move the sequence a little bit and you remove some part of the sequence, you get other stem loop structures. So this folding actually, if you see, you can predict multiple such folds in the RNA, and if you don't really have to change the sequence of it. It just in different situations, depending on the protein that it's binding or on its own in the context it is, where in the cell it is, it tends to fold in a different manner. So it's really not that rigid for RNA as much as it's in the protein. Though it has only 4 things, because it can loop back and forth in different ways, because this stem can be formed like in N-terminal, but there might be a C-terminal sequence of 4 or 5 nucleotides which the run is the same. It can loop back and form a different stem, and then the loops will be different. So the structure is very labile. It's not so driven by the primary. Okay, having said that, there might be, for protein, there might be a few possible structures. In RNA, there are hundreds of possible structures. And again, if the length increases, the domain structure will keep on changing. So as you add sequence, you will change the sequence. So it's often seen in RNA that it can fold in various patterns because of the stem and looping. So it can bulge out like this. As you can see the circles over here, which can bulge out.
Yeah. Last question. As an editor, I have received n number of manuscripts on breast cancer.
Okay.
And everything looks like a case study.
Okay.
How to make it a generalized paper now? It's very difficult for me to go through and see whether it's accepted or rejected because these are all a particular case and a particular analysis and bioinformatics data is given.
Yeah. So so one of the breast cancer things we are seeing is follow-up of the patient data is very, very important because what I would do for the if you do a lot of analysis, you're going to come up with some markers or something or the other, and we really need a number, a very high number, because first of all, breast cancer is very, very variant. There are actually 6 classes. Then we have these personal differences between the type of mutations that you have in there. So there are so many groups and subgroups in breast cancer, and then you're treating them differently. So they fall into more subgroups and things. So each group tends to have a different kind of behavior in the clinic based on its characteristics, based on the treatment, and many other things. So if you have a very small number, then the case study is a problem because it's not going to go through when you have 500 samples or 1,000 samples. So for smaller ones, if you're associating something with disease prognosis or something, it's not going to pan out later. So I don't think that's one of the concerns like we also have. Like if I show something in 5 samples, it's like rejected. I have to go up to 500 and 800 and do a lot of in silico analysis to prove the points that we are making. So that's a problem. To make it better, they need to have N needs to be very large. I think that is true for any study. So does that answer your question, or did I just become very vague?
Which first question or the second question?
Yeah.
No. You want my reaction to the structure sequence relationship or the manuscripts?
Yeah. So, I have to read some of them to know what I would like to see in them actually. So, I don't think I've done a breast cancer patient sample for Journal of Genetics.
So. No. In general, I get many papers on breast cancer, taking 1 or 2 patients, 1 or 2 markers.
Yeah, but I don't want to be, how should I say, 1 or 2 patients is really not going to cut it for me in breast cancer because there's too much variation and too many, like I said, too many subgroups. It needs to be a big study with, or if they can get this one group. You know what I mean? If they go for ER positive patients which have luminal A subtype, they've already typed the subtype of the breast cancer, and then they have 50 samples, then it's okay. Because you're going to interpret everything on the basis of the characteristic of that group, and then that is fine. But 1 or 2, even sequencing data, I will not be very happy with.
Thank you. Thank you, Kartiki.
Thank you. I would like to thank Professor Kartiki Desai for a very nice talk. Thanks very much.
Thank you.